going to hear some very, I think, positive news tonight about some of the directions that this organization is moving in. It was only a few years ago that the CureJM board, in fact, made a determination that it was up to us, up to CureJM, to lead the charge to first identify and then bring to clinical trials new drugs that would be more effective and less toxic to children in treating their JM. In making drug discovery a clear priority, we enlisted some of the brightest young minds on the planet to help us accomplish this goal. And I think equally important, we have invested in institutions with decades of experience in medical research and clinical trials to get us to this goal. Two of those institutions are the National Institutes of Health and Duke University and the Duke Clinical Research Institute, which are represented tonight. NIH has completed one clinical trial in JM for a drug known as baricitinib and is now launching a confirmatory trial in an effort to duplicate the positive results that we've already seen with this drug. Duke, along with CureJM's Chief Scientific Officer, Dr. Andrew Heaton, wave Andrew for us, is leading the charge for a clinical trial in JM for a new steroid that has far fewer side effects than do the steroids in use today to manage JM. I mentioned these bright young minds, and while we have many of them around the country who we have funded through your generosity and fundraising leadership, two of the brightest stars are Dr. Travis Kinder, who has led CureJM's drug discovery program at NIH, and Dr. Kaveh Ardalan at Duke University, who has taken on a lead role in the development of the clinical trial in JM for this new steroid, vamorolone. At the outset, let me say that some of the drugs Dr. Kinder will tell you about are approved by the FDA for other conditions, and some are not approved by the FDA at this point at all. It is possible that some of those approved drugs through clinical trials in JM could be available sooner, perhaps in the next 2 or 3 years, baricitinib being one of them, as these clinical trials prove and come through with positive results in both safety and efficacy for JM. The same is true for vamorolone. It is currently not an approved drug and so is not available for use by patients in any disease. But we're very hopeful that it will be approved by the FDA for Duchenne muscular dystrophy in the first quarter of 2022, and that it will be available on the market for doctors to use sometime towards the end of 2022 if everything goes according to plans for that particular drug's development. Finally, if you have any questions as the presentations are being made, please put them in the chat. But we will also have some time at the end of the presentations from Dr. Kinder and Dr. Ardalan for live questions at the end of the session. And so it's my real honor to introduce to you Dr. Travis Kinder, who will be followed by Dr. Kaveh Ardalan. So Travis, over to you.
Thanks, Jim. And hi, everybody. It's an honor to speak with all of you and give you an update on some of the work that we've been doing that's been funded by CureJM. So I put together a brief presentation to talk about some of these progress. It's going to be fairly brief, but I'm certainly willing for more conversation and questions as you have them. So first of all, I started this project about 4.5 years ago with the seed funding from CureJM. I had recently finished my PhD at George Washington University under the mentorship of a leading myositis researcher, Dr. Nagaraju, at the Children's National Medical Center. And so I was looking for a postdoc position, and NCATS, where I am now, the Institute of NIH, had gotten this funding from CureJM to do JM translational research, and that's what I had been working on for the previous 5 or so years. So it's kind of the perfect fit to to start this project under the head of my current supervisor, Dr. James Ingles, who has a very long history with both pharma, academia, as well as the NIH, including working at Merck and Pharmacopoeia. So if I go to the second slide, please. So I guess first I'll just give a broad general overview of what we consider as translational sciences. So I often like to tell people that NCATS, National Center for Advancing Translational Research, is actually one of the newest centers of the NIH. And our mission is to really focus on this whole translational science pipeline, which we define as the process of turning observations in the laboratory, clinic, and community into interventions that improve the health of individuals and the public. And so this can be thought of as taking biology, basic biology, and other studies into therapies. So our project kind of focuses on the very early stage of translation, where we first, look through the basic research in the field and consult with experts in the field, such as the CureJM Medical Advisory Board. And we really identify some kind of targets that might be druggable. For example, if there's too much of something in a condition, we can try and find things to block it. Or if there's not enough of something, we can try and find things to bring it up. So these are kind of thought of as like drug targets in a condition. Then the next step is to develop assays, which are measuring tools. And you can think of this as having the same root as assessment. So an assay is just a way of measuring one of these kind of biological targets. For example, when you give blood at a hospital and they send it to a lab to measure, let's just say creatine kinase, they're using an assay for creatine kinase. So we develop assays for these targets. Then we use these cutting edge robotic facilities to screen thousands or potentially tens of thousands or even hundreds of thousands of different drugs or chemical compounds in these assays to try and find some that can modulate the assay in a certain favorable direction. Once we find chemical compounds that are active, we can go through a series of medicinal chemistry, which are kind of making modifications of the drugs to try and find the best ones. And then eventually preclinical animal testing in animals, like animal models, and then finally clinical trials in humans. So we've kind of been doing the assay development and drug screening so far. And then things obviously get handed off to clinicians such as Kaveh in the later steps. And the next slide, please. Just a couple of photos of our lab just to get you oriented to what it is we're doing here. So that the top photo is my actual physical lab, that the ADST lab run by Jim, being run by Jim Ingles. And so in this lab, we develop the assays kind of by hand through just traditional biology methods. And then once we've developed the assays, we then put them onto the robotics, which is seen below. And the robotics is essentially just these arms that move the assay plates around from instrument to instrument. So the assay plates are these small kind of postcard sized plastic plates that have a bunch of tiny test tubes in them. And we use 1536 well plates. So each plate has 1,536 tiny test tubes that can each do a reaction. And then these robotic arms just move them around. Next slide, please. So what we've done, as I mentioned, is the very early step of just kind of identifying pathways to create assays. So in consultation with the literature and experts in the field, we've kind of identified there's a growing body of literature showing that there's an abundance of this pro-inflammatory component called type 1 interferon in certain subtypes of myositis, including dermatomyositis, which is the most common subtype in the pediatric population. And so in our effort to kind of make more targeted therapies that are not so broad acting, such as glucocorticoids or immunosuppressives, we're trying to find drugs that can block this specific inflammatory pathway, the type 1 interferon pathway. And this is kind of a cartoon diagram we've put together to show this kind of model that we think is going on in at least one component of JDM, where the type 1 interferon is pro-inflammatory, and so it binds with receptors on the muscle cell, which is this pink cylinder, and it activates certain genes, particularly immune genes. And in this case, the one we're very interested in is called MHC class 1, and it's this purple stick figure here. And what this does is it actually it signals to T cells, which are a white blood cell for autoimmune attack. We've also shown that upregulation of this protein can lead to a further feedback of the type 1 interferon signature, as well as what's known as an ER stress response in the muscle, which causes atrophy of the muscle. So we believe this whole pathway, this whole pipeline of the type 1 interferon and the MHC class 1 is pathogenic in myositis. So our goal is to find compounds that can block this. And if you could advance one. So this shows our goals, blocking this inflammatory pathway. And then the second half of this figure shows one of our methods that we've used to interrogate this pathway. So without going into all the details, we've used CRISPR-Cas9 to genome engineer human muscle cells such that they have a reporter gene attached to MHC class 1. And what this reporter gene does is it actually produces light, much like a firefly produces light, so that we can measure how much of this protein is being expressed very easily and very quickly in the 1536 well plates. So we've used this to, we've attached the reporter to MHC class 1. We treat the cells with type 1 interferon in the dish, and then we add all the chemical compounds to find those that inhibit this pathway, which would represent being anti-inflammatory. We've also developed a whole pipeline of assays to kind of validate this primary screen. And that data is on the next slide, please. So this is just a brief snapshot of some of the most kind of exciting and most promising drugs that we found. To date, we've actually screened over 20,000 compounds, and we've done them all across titrations, which is several orders of magnitude of doses. And in the top right, you can see this kind of waterfall where it shows a bunch of the drugs that we've tested across titrations. And we've pulled out a few here to highlight that are very interesting. And the kind of way to read these graphs is as you look left to right is increasing dose of the drug, and in black is the reporter gene for MHC class 1. So all of these have this S-shaped curve where they come down at the higher dose, which is good. So these drugs are blocking that black signal. And then in the gray, the open circles is a viability of the cell. So we don't want these drugs to be toxic and kill the cells. So this straight line is what we're looking for. In green, headings are approved drugs, and in black are in clinical trials. Most of these, 6 out of 8 of these are JAK inhibitors. So we think that the JAK inhibitor class overall is very promising and should be looked more into. These green ones are approved for other autoimmune conditions, and we hope to be able to relatively easily translate them to JM, easier obviously than creating a brand new drug. And then the final 2 at the end, panobinostat and givinostat, are what are called HDAC inhibitors, which is a whole other class of compounds that seem very promising as well. So I would say these 2 classes, the JAK inhibitors and HDAC inhibitors, seem like they'd have the most promise in trying to initiate clinical trials in JM for these. And then the next slide, please. So this is just a summary of what I've discussed. So we created a collection of assays to measure type 1 interferon MHC in muscle, including CRISPR-Cas9 genome engineered cells. We've screened over 20,000 compounds, including all approved drugs in the US, Europe, and Japan. The JAK and HDAC inhibitors were very effective and most promising for translation. And obviously, I'm not recommending them or anything. They need to be tested in animal models and clinical trials to get FDA approval. And the next steps specifically for what I'm doing is to use this battery of assays for a variety of other experiments and continue experiments, including testing potential environmental triggers of this pathway, which might indicate triggers of inflammation in myositis, identifying brand new drugs and new drug targets, not just already approved drugs. We want to develop new complementary assays, particularly to further explore all these actives, including the JAK inhibitors. And with that, the last slide is just acknowledgements where I'd like to thank everybody who's made this work possible, NCATS, some of our collaborators, including at Children's Hospital of Philadelphia, who's also getting funding from CureJam, and obviously the CureJam Foundation, and all of your tireless work and everyone's contributions, and the patients and family. And with that, I'll take questions or let Kaveh go next.
So as Travis was mentioning earlier, like I'll kind of share with you guys a little bit about what happens on the other side of that translational arc, where we bring new medications to patients in the context of clinical trials. And specifically, I'll be talking about vamorolone vamorolone and its role potentially in juvenile myositis. So next slide. So just a little bit of background, and for this audience, a lot of this is probably beyond well known, but just to kind of make sure we're all kind of on the same page. I mean, steroids are the double-edged sword of caring for kids with juvenile myositis. So we know that way back in like the 1940s, 1950s, when it was sort of the pre-steroid era, no one really knew at all what to do for kids with myositis. This was a really highly deadly disease. I mean, almost about a third of kids would actually succumb to this illness. And so in that sense, steroids have been a revolutionary treatment when they first started to kind of be used more widely in the mid to late 20th century. But we know now that like high dose steroids are needed to kind of get both adults and kids with myositis into remission, and that it often takes a while on steroids to really get this disease into remission, often well over a year, sometimes even over 2 years. So definitely a lot has hinged on the role of steroids, but at the same time, like they're kind of a medication that we lean heavily on, and there's some challenges with that. So next slide. I wonder if the slides are advancing. Okay, perfect. And I can also share my screen if that's easier, whatever that works better from a tech standpoint. Okay. So I'll pick up from here. So again, folks on this call will know that steroids are, basically the linchpin of treatment. So about 2/3 of kids with JM will have a relapsing or chronic course of their illness. So it's not necessarily something we can expect that kids would simply outgrow. And although treatments more broadly have improved, like we know that methotrexate works, we know that rituximab can be helpful, we know about other medications that can be helpful. Even still, with all these advances that we've had in the last couple of decades, we find that a lot of our patients still end up on lengthy courses of steroids. There was a recent study from the NIH actually that's found that at about a decade after diagnosis, patients with JM about half of patients with JM, were still on at least some steroids. That doesn't mean they were on for a decade straight, but it just goes to show that somehow that it's almost like being kind of on this roller coaster where you're on and off steroids or on fluctuating doses over time. Next slide. So, and then of course, everyone on this call also knows that steroid side effects are common, and that's an understatement. So there's a huge range of side effects associated with steroids. We know that they weaken bones. I've had a couple of my patients for sure have like fractures of different parts of their body, sometimes with pretty minimal impact. Almost universally, there's weight gain, sometimes really intense amounts of weight gain associated with steroid use. Slows down growth, so there's kind of stunting where kids don't really get as tall as they otherwise would have been without steroids on board. Can also delay puberty. Steroids are immunosuppressive, and they're associated with risk of infection. I think something that those of us who are physicians underappreciate, but I think parents and kids know better than anyone is the mental health impact of steroids, the kind of fluctuating moods and irritability and depression and anxiety and so on that a lot of kids can experience is sometimes exacerbated when they're on steroids. And this is just a partial list. I mean, we could kind of keep going on. I think the bottom line is we hear loud and clear from our patients and their families that we need better treatments, and that although steroids are life-saving, there has to be something better out there for our kids. Next slide, please. And I think I see the chat really lighting up. So yeah, we've seen this. So then, in terms of alternatives to steroids, I think one thing that's been really exciting is looking to other rare disease populations that also require a lot of steroid use and seeing if there might be opportunities to learn from those communities, learn from the researchers in those communities and the families they're working with. So there's a condition called Duchenne muscular dystrophy. This is not autoimmune in the sense that JDM or JM is, but it is a muscle disease, and there is inflammation in the muscles of kids who have this condition. So it's a rare genetic disorder. It affects boys, and it does require long-term steroid use because without steroids, there's kind of loss of muscle function and eventually loss of the ability to walk and to get be functional. And as you could imagine, for a condition like this, where steroids are kind of a major treatment and basically on board for years at a time, these are patients who also experience a lot of the side effects of steroids. And so folks in the Duchenne muscular dystrophy research world and the families that have advocated for better treatments collectively have thought, well, what if we could find a way to get all the benefits of the steroids, but separate that from the side effects? So next slide. So, so then there's some interesting work that was done over the years to kind of see if there's some way to pull apart the good and bad of steroids. And without going into a ton of detail, what you should know is that in the body, there are these structures called glucocorticoid receptors. And these are pretty complex structures. When you take steroid medicine, the steroid molecule, whether it's prednisone or a different steroid, it interacts with these glucocorticoid receptors. And because the structure of these receptors in 3 dimensions is pretty complex, there's different places that steroid compounds can kind of connect to the receptor. And when it connects to different parts of the glucocorticoid receptor, you get different effects on the body. Some research over the years has shown that basically there are some sections of the glucocorticoid receptor that are predominantly involved in the anti-inflammatory effect of steroids, so the good stuff. And then there are other sort of sections of the glucocorticoid receptor that are responsible for most of the side effects, so the bad stuff. And so the thought was, what if we could create a molecule that would stimulate mostly what you want from the glucocorticoid receptor and leave alone the part that brings on all the side effects? And so vamorolone is the first compound that has been studied actually in patients to kind of accomplish this aim. So it's being called a dissociative steroid because the idea is it kind of dissociates the benefits of steroids from the side effects by activating mostly the anti-inflammatory segment of the glucocorticoid receptor. Next slide. So, as Travis was pointing out, there's a long path though from seeing in a lab, in cells or in a mouse model or these kinds of like lab-based approaches. There's a long road from kind of bridging from what you see in that setting to actually proving that in real patients who are living their lives, that this medication actually lives up to the promise. Well, so far there have actually been some encouraging results from trials that have been conducted in Duchenne muscular dystrophy. So there have now been phase 1 and phase 2A, 2B trials conducted. Phase 1 and 2A data is published, and phase 2B is has been presented. At least top line results have been, and publication should be following. But basically, so far it looks like when kids with Duchenne muscular dystrophy receive vamorolone, if you compare those kids to those who receive prednisone or prednisolone, that the vamorolone seems about as good or sometimes actually better for preserving muscle function for those kids compared to traditional steroids. And the really nice thing is that the side effect profile does seem to be milder with vamorolone. So kids are growing, they're actually getting taller the way they should. whereas the kids on prednisone are still having the growth stunting. There are some blood tests that are being done to look at the health of the bones to see if maybe that bone fragility can be avoided. And so far, those studies seem to be suggesting that bones should stay stronger and healthier over the long haul with vamorolone. There's some early indication that behavior and mood are actually not nearly as affected, may not even be affected potentially by vamorolone. So that's, I think, super exciting. And although we're hoping that weight gain can be completely avoided, it does seem like if you push vamorolone doses pretty high, you'll get some weight gain. But in general, it doesn't seem to be as intense, and it doesn't seem to be as universal as with traditional steroids. It's kind of a little bit more variable. And they did notice that if they brought the vamorolone dose down, there wasn't as much weight gain, or you could actually reverse that weight gain. So still seems somewhat favorable in that regard. But there are a lot of unknowns still for, oh yeah, go ahead, next slide. Thank you. There are still some key unknowns though, and we, even when vamorolone is approved for Duchenne muscular dystrophy, we would need to answer some very important questions before we would know if this is right for our kids with JM. So a couple of those questions here. So the first is, does JM require higher doses of vamorolone? You know, Duchenne muscular dystrophy, like I said, there is inflammation in the muscle of children with Duchenne, but it's not nearly the kind of intense inflammation that we see in juvenile dermatomyositis and other forms of myositis. So, and it's not systemic inflammation to the same degree as with juvenile myositis. So we're thinking that maybe our patients with JM will need higher doses of vamorolone. And if that's the case, what is the right dose? We need to make sure that we're tailoring things appropriately. The second big unknown is, what's the impact of the other immunosuppressants, taking that in combination with vamorolone? You know, most kids with Duchenne don't have the kind of really complex multi-medication regimens that kids with juvenile myositis have to take. And, you know, when you think about, say, methotrexate plus a new medicine, or, you know, cyclosporin plus a new medicine, or any number of different meds that kids with JM take, we need to be careful about kind of the mix and match of these medicines and make sure that this is tolerable, that's appropriate. And then there's some questions about age and gender. So as I mentioned, Duchenne muscular dystrophy affects boys and young men. Juvenile myositis can affect boys and young men, but predominantly affects girls and women. So we need to think carefully about, are there gender differences in how this medication will be working? And then there are age differences. So Duchenne muscular dystrophy presents pretty early in life. Most of the kids who've received vamorolone in trials have been between like 4 and 7 years of age. So we know that our kids with JM experience symptoms through a broader age range. It's, you know, relapsing and remitting kind of conditions. So we need to make sure that whatever doses we try out in a trial setting are going to work across a broad age range. Next slide. So how do we bridge this gap? So the first thing is to just say thank you to CureJM, thank you to all of you on this call, because we cannot make progress on this without the kind of generous support that every single person on this call has provided. So that's first and foremost. And CureJM has been really, really generous in their support of now a year-long effort, a collaboration between myself and a team at Duke University, as well as the Duke Clinical Research Institute, which is a very large organization based at Duke for conducting large multi-center clinical trials. In fact, the largest clinical trial in pediatric lupus, another rheumatic disease, was conducted through the DCRI by Duke faculty. So we've got a good team here. So collaboration between folks at our site, Andrew Heaton and others in CureJM have been super involved, and then the company Reveragen Biopharma that you know, has vamorolone and is doing the studies in Duchenne. And over the course of this year, what we've done as a group is really put our heads together over many meetings, engaging with experts in clinical trial design and statistics and regulatory aspects of clinical trials, to try and figure out what would a clinical trial in you know, juvenile myositis look like. And the process basically involves kind of drafting a sort of a synopsis of what a trial might look like, and then requesting a meeting with the FDA to review that synopsis and see what the FDA thinks. Do they agree that this is an appropriate approach, or do they have any concerns? And then once you have that meeting with the FDA, you get regulatory feedback from them where they're basically telling you, if you really want to study this properly, we would recommend adding or subtracting the following components for your trial. Next slide. And so, so far, just kind of broadly speaking, what we're proposing, and we're gearing up to send this to FDA for their feedback, is a trial where we would actually have both adults with dermatomyositis and kids with dermatomyositis in the same trial. And the reason for that, it's multiple. So one is, these are actually similar enough conditions that we would have a much better time getting a good number of patients if we expanded enrollment across the full life course. And there's precedent for this. So the study called the RIM study, which established rituximab as an effective treatment in myositis, was it's the largest trial to date in myositis history, and it enrolled both adults and kids. That also speeds up the process of getting medications to children, because a lot of times, understandably, it's important to first make sure that a new medication is tolerable in adults before giving it to children in a trial. So our approach would be to enroll adults and children with dermatomyositis who have active skin and muscle disease that requires a steroid of some kind. Enroll some adults first. Once we have enough adults in the study, do like kind of an interim safety analysis. Just make sure that those adults are doing okay on this medicine. And if we see a good signal there that it's safe to proceed, then we would open it up to children to enroll while we continue to enroll additional adults. And the comparison would be between vamorolone and prednisone. We would compare the efficacy, so how well does it actually control symptoms of myositis and get rid of inflammation. We would, of course, look at safety and side effects, because our hope is that that's really where we'll see a massive difference. And then, again, because we're going to need to know what the right dose is, we'd have to assess multiple doses, including maybe pushing for higher doses. And the other thing I think is important to highlight is we want to make sure that there are some measures from patient and parent perspective about physical function and emotional well-being, because we need the voice of our patients and their families to be incorporated into trials. And that's something we've been thinking about. Next slide. So these are the first steps on what is going to be a long road ahead. But what we have accomplished so far is getting, I think, really the right experts and the stakeholders that we need to start thinking carefully about how to design this trial. We now, within our team, Duke, CureJM, and RevereGen have kind of a broad consensus about what the study design we think should look like. And so we hope to incorporate feedback from the FDA. Once we have that kind of regulatory feedback, we should be able to then start thinking in greater depth about feedback from patients and families. And then once we've got all those ducks in a row, it's time to get funding and get trials going. And next slide. Just want to thank every one of you again for all you've done. You guys provide us so much support, so much energy, and we hope that we can provide you and your families hope and better treatments. So thank you so much.
A couple of questions about how would a kid sign up for the clinical trial?
That's something that will come in the future, but basically, typically with these trials, because dermatomyositis and other forms of myositis are rare, we have to do what are called multi-center trials. And so part of the trial planning process is once we know what the study should look like, once we've got FDA feedback and we have a clear vision that is agreed upon with FDA about how to proceed, that's when we'll start identifying, okay, what are the different hospital systems that we would work with to offer enrollment in this study. And basically, it would be a matter of, I'm sure we would partner with CureJM to get the word out as well. So, if you're plugged into CureJM, you will know when this study is live and where to enroll. And the hope would be that there's somewhere close to where you are if we can get enough centers involved. And then you would just let your doctor know that this is something you want to do. They would connect you with one of the study teams, and then you meet with the study teams that are offering this type of trial, and they would go through risks and benefits with you and make sure you really feel fully informed before you decide to try it out. Because remembering that while we're very hopeful, technically we don't know yet if this is a better medicine. And so, they would want to make sure you feel fully educated before you make any decisions.
One other question in the chat for you, Dr. Ardalan, from Sari is, do you have a timeline of when the FDA will get back to you with feedback?
Yeah, it's an excellent question. So we actually are gearing up to submit our request for a meeting with the FDA in the coming weeks, so this month. We just got some kind of word from our partners at ReveraGen, like they've gotten feedback from trials in other autoimmune diseases that they're proposing. And so we just got some new information about kind of one of those trials and what FDA thought, and we're going to be meeting as a group, CureJM, ReveraGen, and Duke teams to kind of think about, in that setting for these other autoimmune diseases, is there anything we need to look to incorporate into our approach to myositis before we reach out to FDA? Because you really want to have, I mean, you have this sort of one shot to get that feedback, so we want to be really ready to get it. But in terms of timeline, we're hoping to have that request submitted to them. And then obviously they're very busy, so it may take a good number of weeks to get the meeting, and then a couple more weeks to get the feedback. But I think the short answer is soon, very soon, next couple weeks to months.
Is there a current alternative to steroids and methotrexate that you can recommend?
Unfortunately, there's not really an alternative to steroids. And that's actually both why this is such important work, and why we're really grateful to CureJM for supporting this work, because I don't have to tell you guys, like, this is an audience that gets it. We need an alternative to steroids. It's great that, it's a life-saving medicine, but it's not great that it does all this other terrible stuff. And so, for kids who truly need steroid medicine, there's not really an alternative. Now what there can be strategies to combine steroids with multiple other immunomodulating medicines. So a lot of times, if I have patients who have had a lot of steroid side effects, or where I'm worried that they'd be at risk of steroid side effects, my approach will often be to, from the get-go, when I'm starting treatment for a new disease or for a flare, think carefully about, do I just want to start methotrexate and steroids? Or since this is a patient who I might need to get off steroids even quicker, maybe I should add IVIG more quickly. Maybe I should add other medications more quickly, because sometimes combination therapies of other medicines that are not steroids help you potentially to get off steroids quicker. But it is a rock and a hard place that we're sometimes caught between, when we have steroid side effects to deal with and the disease to deal with.
A family who, I believe this must be their myositis antibody, anti-NPX2. And they did not have luck with tofacitinib or baricitinib, and are wondering if there's other alternatives to those JAKs.
Yeah, I mean, I can speak a little to that, but Travis, maybe, I don't know, because you've got some interesting investigational data, so maybe you have a sense of what the future holds. I don't know if you wanted to go first.
Yeah, I can make a brief comment. So our assay system it's very simplified, so it doesn't really take into account antibody status. It's just a normal cell, a muscle cell treated with interferon. So we're basically just looking for drugs that would be very good for blocking the interferon pathway. Unfortunately, for this specific example, if these other two JAK inhibitors were ineffective, it's probably not likely that another JAK inhibitor would. It kind of suggests that maybe, so these drugs are very good at blocking, for example, the interferon pathway, but if it didn't help, then it may be that there's a different pathway going on in your specific case and not the interferon pathway. So as we know, this condition is very heterogeneous. So different people can have different factors contributing. On a kind of positive side is that maybe sometimes it's good to know what doesn't work, for example, and to try different things. And I don't necessarily have a better answer than that. I mean, we're very much looking for interferon drugs. So our specific work may not be as helpful for this specific case, unless there are, it may just be that the interferon signature wasn't as dampened as it could have been, in which case our studies would be helpful, finding better JAK inhibitors. But that's kind of more of a clinical thing that I think Kaveh could probably address.
Yeah, I think those are really important points, and this is one of the challenging things about dermatomyositis and other forms of myositis is just every patient is different, and there's not a one size fits all approach. There are, as Travis was showing, there are other JAK inhibitors, out there. Tofacitinib and baricitinib are the ones that have really been best studied so far in juvenile myositis. So my sense is that if neither tofacitinib nor baricitinib worked, I would kind of wonder if maybe a different class of medications besides JAK inhibitors might be the way to go. And so that might be a worthwhile conversation that I imagine you might already be having with your child's doctor. Another question I would have is, did it simply not work at all for either skin disease or muscle disease, or was it a situation where, as we sometimes see with our patients, one component of the disease improved and one didn't? So sometimes we have patients whose skin rashes are really resistant to treatment, but their muscle disease improves. These kind of nuances are worth exploring because depending on the specifics, sometimes you can kind of decide, like, should we move on entirely from JAK inhibitors? Should we add something to a JAK inhibitor? And if we're going to move on, what should we move on to? Yeah, and there are a lot of other medicines besides JAK inhibitors, and they can be given in combination therapies. So that's the other thing, is it's not always the case that just one medicine at a time is the right way to go. Some kids need more than one medicine to get into remission.
And Dr. Ardalan, I think this last one is also for you as well. Since this particular patient is using medications off-label anyway, do they have a chance to use vamorolone off-label, or when would that sort of thing be possible?
Yeah, so vamorolone's not approved right now, so there's no way to currently receive it outside of the setting of a clinical trial. But my understanding is that ReveraGen is going to be putting in an application for FDA approval of vamorolone in Duchenne muscular dystrophy this year. So as was mentioned earlier on the call, it seems likely that vamorolone is going to become available to kids with Duchenne muscular dystrophy. If I'm being very honest, I would probably say I would not recommend taking it for juvenile myositis just yet, though, because as I mentioned, there are some real unknowns. Like I said, we don't really even know what the right dose is for kids with myositis. And if we underdose kids with myositis, that could be potentially really dangerous. If we overshoot, that could also be dangerous. Although vamorolone seems like it has a better side effect profile, that's in comparison to pretty low doses of prednisone that kids with Duchenne muscular dystrophy are on. We haven't yet done a comparison between kind of the higher doses of prednisone that kids with myositis have to take and then potentially higher doses of vamorolone that might be necessary. And so I would say your doctor wouldn't really even know what dose to give. I wouldn't even know. That's why we'd have to do a trial to answer that kind of question. And as I mentioned, we don't technically know how it works or how well it works and how safe it is in kids who are on multiple other immunosuppressants. So I think for now, this is not something I would try just this minute, but we're going to do our level best to keep this process moving as quickly as it can.
All right. Well, Dr. Ardalan and Dr. Kinder, thank you so much for your extraordinary presentations. As Shannon mentioned, there have been lots of thanks scrolling through in the chat window for all that you have done. We appreciate your partnership and your institution's partnerships and the direction that you have helped take our program, your program, really, in drug development for better treatments and one day a cure for juvenile myositis. Thank you very much. To all of our families, I know many of you have already signed up and are planning to participate in the most important fundraising campaign ahead of us in the immediate future. This is our CureJM's Holiday Challenge. I want to thank all of you for signing up for that and for creating your pages. It falls on Giving Tuesday, November 30th. We always feel a bit like we kind of own that day, if you will, as a small nonprofit that has been raising money around the Holiday Challenge now for any good number of years. About $3 billion will be given to charity on Giving Tuesday. And our goal, as it is year in and year out, is to assure that CureJM is just a part of that $3 billion in human generosity and philanthropy that takes place on November 30th, Giving Tuesday. So the best way we can accomplish this is simply to spread the word about JM and to tell your story and tell your story on our pages, our Holiday Challenge pages. You can do that if you have not already signed up. You can sign up by going to curejm.org/HopeHero, or you can message Shannon Malloy. Everybody, I think, knows Shannon Malloy on this. And if you do so early, you can also get one of these great Hope Heroes t-shirts that many of us are wearing as we move forward. As I mentioned, no organization in the world that I'm aware of has done more to advance research in not just juvenile myositis, but myositis overall than CureJM in our 18 years of history. We have funded some $18-19 million in grants over this period of time. We currently have a grant portfolio that totals about $5 million and funds programs like the two that you've just heard about tonight. This is all possible through the success of our Holiday Challenge, among other fundraising programs. And it's all possible because, as I mentioned at the outset of this call, we are a family of families, and we are an extraordinary grassroots organization, and we know how important this is to parents, to grandparents. Thank you, grandparents, who are also on this call, for us to find these better treatments and ultimately a cure. Thank you, Kaveh, for your leadership at the CureJM Center of Excellence at Duke University. Thank you also, Travis, for being such an extraordinary partner. One of the things that we've always needed to do as a small organization is to leverage our investments with large, highly accomplished institutions like Duke and NIH, and we're truly honored to have you as partners on our team. With that, I want to wish everybody good night. Here we are right at the 9 o'clock hour here on the East Coast. And if you have any follow-up questions at all, if something comes to you in the middle of the night or in the next couple of days and you want that question answered, please feel free to email me, james.minow@curejm, or Shannon, shannon.malloy@curejm.org. And we'll be happy to either answer your question or get it to the right person who can do that. Thank you all for being with us. Look forward to chatting with you more in the coming months, and certainly in the month ahead, leading up to November 30th. And if I don't see you before the holidays, please, everybody, have a wonderful Thanksgiving and a wonderful holiday season as we move to what will, I'm quite certain, be the most promising and productive 2022 ahead of us. Thank you all. Good night.